Okay, so our next speaker is one of our friends from UH. It's Russell Galante, and Russell is a junior extension agent for ornamental crops and landscape industries based on Oahu. He did receive his master's degree from CTAR. He's worked with the green industry for 17 years. In his current position, he's working on a number of projects related to pests and diseases in the green industry, new temperate cut flower trials, lowland protea variety trials, new tree variety releases, as well as working on new marketing opportunities for nursery growers on Oahu. So welcome, Russell.
Thanks for having me up here. So I started about 6 months ago in April, and immediately I moved over from Komohana here in Hilo to Honolulu, and the first thing they said was, we've got this ginger problem, this red flowering ginger problem that you need to start work on immediately. We've got a grant for it, and it's the most pressing issue for Oahu's floriculture and nursery industry, and so I made sure to incorporate and envelop the landscape industry into this problem as well, because this disease issue is island-wide. The first issue is that we've got virus infections in flowering gingers in Hawaii, and who I'm working with on this project, on this portion of the project, is mainly John Hu, and then all of his laboratory technicians, as well as Mike Melsner and Jerry Sugano, extension agent on Oahu, who took on the grant originally. So what was happening about 5 years ago, before I was involved, growers started to report that they were having quick decline in ginger in the Kahului region of Oahu, which is where that red circle is. So it's an incredibly rainy area, and it's also was an epicenter of red flower ginger production at the time. And so that was in about 2014 when these individuals started reporting that there was decline in red flowering ginger production due to fast decline symptoms related to just leaf dieback, stalk dieback, and lower production.
So do we have cut flower ginger producers in the audience today?
Yeah.
And you're doing red ginger? Okay, great. And hopefully there might be some people online? Maybe. So the problem has since spread island-wide. We've had reports through most of the ginger growers throughout the island, as well as doing surveys on Oahu. It's within the landscapes in the resort areas, as well as residential areas. It's good to hear my own voice. I just got told I have a nice radio voice, so I'm feeling really good about myself. Sorry. So the symptoms that we're looking at are going to be mottling, streaking of the leaves in terms of virus. So I'm first talking about the virus. And all of this work I'm presenting on behalf of John Hu, Dr. John Hu, who is a virologist at UH Manila. So he's doing all of the virology work. There's a lot of people working on this problem who I'm going to also discuss later. But first of all, we're talking about John Hu's work. So there's multiple symptoms, either mottling, mosaic patterns, or a combination of the two. And it's all the way down to looking like just chlorotic growth. So a lot of the growers at first thought they just had nutritional deficiencies. There's also reports of symptoms on flowers. So speckling, scratching, and streaking on flowers is related to some of these disease symptoms. So in Hawaii, two viruses were originally determined for infecting red flowering ginger, and they were banana bract mosaic virus and canna yellow mottle virus. And these are both viruses that were jumping off of other plants, banana and canna. So for banana bract mosaic virus, which is going to be the BBRMV in the future slides, you're seeing the symptoms of the mosaic patterns as well as mottling on the flowers. And for canna yellow mosaic virus, we're seeing slightly less modeling symptoms, and they are exhibiting more of that chlorotic growth. So what growers were thinking was either nitrogen deficiencies or something along those lines. Now these two were identified as being viruses that were infecting gingers, but they weren't necessarily causing the symptoms that were alarming growers or the industry, which was stunting. So banana black mosaic virus is a podovirus, and canna yellow mosaic virus is a badenovirus. We do know the vector for the BBRMV, and that is it's an aphid, Pentalonia. And for the canna yellow mosaic virus, we don't necessarily know what the vector is in Hawaii. Now for other badenoviruses, it's generally mealybugs and sometimes aphids. But we have not seen an actual insect vector or been able to determine a vector for CHIV. And so here's just what the viruses look like, as well as what their vectors would look like. So aphids for BBRMV and the assumed mealybugs for CHIV. So where we really start to see issues is with co-infections. So we started to find multiple viruses within one plant. Now the decline symptoms and the stunting were not related to either of these viruses infecting the plant individually, but when you saw co-infections of BBRMV and CHYMV, you started to see some stronger symptoms of stunting, of reduced yields, and of plant decline. So if it was complicated now, it gets worse because we started to find new viruses. And one of them was bean comic mosaic virus. So this is the first time or the first report in the world of bean common mosaic virus infecting gingers. So we were able to report that. This was the only time somebody had seen BCMV and reported on it in ginger plants. And in this case, it was actually a co-infection of canna yellow virus, mosaic virus, and BCMV, bean common mosaic virus. And then, of course, to make things worse, another virus was identified, and that was banana streak virus. And so the only symptoms we've seen from banana streak virus were this streaking and the scratching of flower petals on the ginger plants. We actually haven't found that many plants that have been infected by BCMV or BSV. So far, most of the infections have been canna yellow mosaic and the banana black virus. So the conclusions for the viral infections were that there's currently 4 viruses that are infecting gingers as we know it. These are pretty common viruses in Hawaii, so they've been noted on other islands. So in terms of quarantine, that might be something to consider if we were thinking about quarantining Oahu plants. It's most likely that these plants are already, or these viruses are already on other islands. The big issue is that what Lisa described of that plant environment host interaction, or the pathogen environment host interaction. They're not getting all 3. So beyond viral symptoms, the problem is there's the virus and other pathogens. So there's stunting and weakening of plants, but beyond viruses, there's another pathogen actually causing the quick decline symptoms. And that pathogen is unknown. So when this originally came out, the Department of Ag had sampled some of these plants, and they found a number of fungal pathogens as well as an unknown bacterial pathogen. And so the assumption was it was going to be a bacterial or a fungal pathogen that was giving these plants that double knockout punch. They would get sick with the viruses, and then they would get killed or knocked back severely to a point of being beyond a threshold of production that was useful for growers. So the symptoms of these diebacks was, like I was saying, stunting and dieback, first of leaves and then of stems. Plant height usually decreases up to 6 feet, so you're used to seeing red flower and ginger at like 10 feet.
something like that. Instead, most of the fields that we're dealing with are below our knees or below our waist. So these growers' plants have reduced in size significantly, as well as flowers being reduced. So instead of 6 to 12 inch flowers, they're having very small flowers between about 3 inches, very thin. It looks like flowers that are produced in California. That was a jab at California. Yeah, and the streaking and scratching symptoms, but that's viral related. So in terms of the initial symptoms, you'll start seeing yellowing of the leaves, especially with leaves that are recently unfolded, as well as tip dieback. And so it starts to look like a root rot issue. And there's actually also root dieback that's occurring, but this is just above ground symptoms, yellowing, curling of the leaves, and then eventual whole stem dieback. And something we've noticed on all the plants is the dieback of roots, as well as actual symptoms of fungal pathogens on the plants. Now, whether or not these are secondary infectors or the pathogen that we're looking for, we're not 100% sure of. Where we're at now is that we had done sampling on Oahu. Samples were given to the bacteriology lab. Let's see if that's my next slide. Okay. So samples were given to Dr. Arif's lab, Mohammed Arif, at UH Manoa campus. They tested for bacteria, and they were also given to the laboratory of Dr. Janice Uchida. So unfortunately for us in this project, Dr. Uchida had just retired. She retired a couple months ago. Luckily, her brother and lab tech, Chris Kadoka, is staying on until December, and he's helping me with any of the fungal work that we're doing. But we're going to be in need of a mycologist come December or January. So, you know, if there's any USDA folks that want to help me out, I'd appreciate it. Anyways, Dr. Hu also got the samples, and he was testing everything for the viruses and potentially additional viruses. Now, the samples that we got back, the results that we got back from Dr. Arif was there were no known bacterial infections that were on them. So we're thinking that it's not bacterial. And we found a number of fungal pathogens on the samples that we submitted, and I was working on Dr. Uchida with. So the current understanding is that we've got several viruses. We know that. Viruses reduce the vigor of the plants. Dr. Hu likes to relate it to something like HIV, where it's making the plant sick, and then that plant is more susceptible. It's like an autoimmune disorder to other plants, to other pathogens. No bacterial pathogens were present. Fungal pathogens were identified. Common pathogens that were identified were Phomopsis, Phoma, Fusarium bipolaris, and several others that we don't have ID yet. So there was no marasmius in any of the samples, so we're thinking that marasmius is not the fungal pathogen that's causing the quick dieback, which it would have been, I think, a little bit more simple if it was actually marasmius. But we have to figure out which of these guys is the issue, if they're the issue, and whether or not they're an issue with or without the viruses, and which viruses are the most damaging when you add the fungal pathogens. So right now we're at the point where we had, using Robert Kading, surveyed some of Hawaii Island, so I'm not sure if Shane, if Robert had worked with you at all, getting samples from you or looking for symptoms. So he was surveying folks on Big Island, asking if they had seen symptoms like this and checking plants for pathogens and virus. We were able to determine a few plants that we assumed are clean, and Robert ended up, before leaving, giving me those plants. So I flew over, picked up the plants, brought them back to Manoa. So we have hopefully clean ginger stock that we are going to do pathogenicity testing on. Now, when we do that, John Hu is also going to test these plants for virus. So we're at the point where November 6th, I'm going to work with Chris and do pathogenicity testing with those fungi that we have incubating. And then we're trying to identify best management practices for controlling the virus vectors, as well as assuming it's a fungicide, we are going to do fungicide trials for the fungal pathogens. So before understanding the problem as a whole, we're still trying to look at educating growers on proposed management, assuming that we've got the virus and we've got fungal pathogens. So we're using the general idea of using integrated pest management to manage these disease symptoms. And of course, for the virus, you have to work on prevention, making sure that you're preventing the vectors from entering your system with the virus itself. And of course, cultural, physical, on up to chemical. So for you folks on Big Island, the big thing is making sure you don't get the virus in your fields. So not bringing plants back from Oahu, not bringing in unknown or possibly dirty ginger plants, and making sure that you are keeping yourselves clean, your equipment clean, and your vehicles clean if you're visiting other farms. Now, some of the viruses can be spread mechanically very easily, some of them can't, and fungal pathogens can be spread mechanically easily, so they don't require a vector. So you want to make sure that you're clean as well. I think this is an interesting picture. So this is at Lion Arboretum. I was running around trying to find pictures of other cultivars of gingers to recommend to the landscape industry and to possibly cut flower growers. So this ginger in front has virus, and it's got the stunting symptoms. In the back, about 3 feet away, you've got a ginger that is its neighbor, and it has not been infected. So in terms of prevention, there's almost, you can see that it doesn't matter what you do. Sometimes it can be right next to it and you're not going to get the infection. But for growers of cut flowers, you want to make sure that you're starting clean and you're inspecting your plant material for symptoms before you're using. If you're finding any plant material that is infected, you want to remove it. For cultural, you want to make sure that you're maintaining healthy plants. We discussed this in another talk. So ginger loves water. Ginger can be fertilized and it's a heavy feeder as long as it's not infected. But as soon as these plants are showing these viral and fungal symptoms, excess water and excess fertilizer are exacerbators of the issue. So high nitrogen will encourage aphid and mealybug populations, and it can promote fungal growth. So CTAR recommends a 1:1:1 or 3:1:5 NPK fertilizer. Now that's pretty low for a cut flower grower, and if you're infected, you still want to try to do something around there. So only anecdotally have we seen success in fungicide trials. So we've got a grower in Kahuku, and this is his field in that picture. He has the virus, he has what we assume is the fungicide or fungal pathogens. He successfully controls it using Manzate. He's used a few other chemicals, some of them not so labeled for cut flower production, and the only one he's had success with is Manzate. So we're looking at Manzate as well as a number of other fungicides for chemical treatments, including Heritage, Dithane, and copper sulfate. In terms of insecticides, we're not recommending broad-spectrum insecticides for control of these aphids or mealybugs. It's a non-persistent virus, so these aphids can infect the plant before you even know you have an aphid infestation, and then they can not host the virus anymore by the time you find them. So if you're spraying for aphids with a broad-spectrum insecticide, you might be eliminating predatory insect populations. So if you find aphids, using a good scouting program, you would want to spray with horticultural oil or insecticidal soaps. And if you have populations that are too high, then you might want to consider broad-spectrum insecticides. And if you have infected plants, you want to destroy them and replace them. For these folks that are in Kahalu, the farms that are really badly infected, they would have to basically start over. But if in your fields you've got 1 or 2 plants that are showing symptoms, you want to remove them, make sure that you don't have aphid populations or mealybug populations, and replace those plants with another clean ginger stock. And there are some alternative species that we're not seeing the disease symptoms on. So some of them are cultivars and some of them are straight other species. So this was kind of for the landscape industry, but it might also be
helpful for cut flower growers, because in the epicenter of where the problem was, we were seeing other Alpinia purpurata growing that did not exhibit any symptoms, and that was Jungle King and Raspberry. In fact, this was at Ho'omaluhia. This is a stand of the Raspberry, Alpinia purpurata, while other red flowering ginger were showing all symptoms and they all had decline. These plants are perfectly healthy. So that's a testament to the genetics behind virus and fungal resistance. And of course, we've got Eileen McDonald and Tahitian also not showing symptoms on Oahu. Here's just a few other. These are at Lion Arboretum. So in conclusion, we've got 4 viruses causing symptoms. Co-infections also occur between the viruses. Fungal pathogens are the most likely cause of the quick decline in the ginger stands. We're determining management strategies and trying to survey for statewide understanding of where the disease is at. So Robert did a little bit over on Big Island. I think I can come in and do a little bit more of surveying for growers. We need to work on Kauai and Maui Islands as well. I just want to talk about one other thing because it might be helpful for growers over here. On Oahu Island, Podocarpus Blight is an issue that HDOA brought up with me, and it's something that I'm working with Manco at HDOA on. So this is not grant money, this is just something that the landscaping industry was worried about, and Eric asked me to keep you guys updated because Podocarpus is potentially a good cut foliage, a plant, and also a lot of growers use Podocarpus as windrows or windbreaks. So we found 3 pathogens on this Podocarpus that are causing the blight, and it's Alternaria, Pestalozza, and Coletotrichum. And we've done pathogenicity testing on them, as well as some studies on what's causing them to germinate under certain conditions. So really healthy Podocarpus aren't getting the blight. Podocarpus mainly close to the ocean, so in landscaped areas that have a lot of salt damage, we're seeing a lot of this infection. And we've also noticed through a little bit of experimentation that sun damage, the salt damage, and mechanical wounding is causing the germination to occur of these pathogens. And also germination almost always was only occurring when in vitro experiments when we were using plant exudates. So we were grinding up leaves and putting them into the media. And the germination would not occur unless those exudates are there, which is also an indicator that the plants are needing to be damaged in order for germination to occur. So we're at the point where we're going to also do fungicide trials on this Podocarpus. Yeah. So that's all I have. 6 months in, I need to do statewide surveys. If you are a grower, you want to contact me, I would love to come sample your plants. And of course, take questions. Thank you.
So you mentioned that you don't see the ginger virus here, but you feel it may be spread to the neighbor islands now?
That's from John Hu. Him and I were talking about it, and he says that viral symptoms these viruses are most likely on other islands because they're very common on Canna. Now, BBRMV has not necessarily been a problem for bananas in Hawaii, but it's probably on bananas. And that was John Hu's recommendation was, I don't know if we should work on a quarantine program. It might not be helpful because the viruses are most likely already there.
Yeah, our experience with any kind of quarantine is it takes a long time to do. Unless it's something really, really highly important to the community. So for the growers here, are you going to have something published?
Yeah, so once we have something useful that's going to say, okay, this is an actual control that we know works for this, we're going to have it published. Yeah, and that's part of the grant is developing the best management practices. So first things first is we need to figure out if mealybug is the vector of the CAYMV and what the other vectors are of the other viruses, and then working on control for them. And so that's mainly going to be using other research because we're not going to be able, with the money that we have, unless we get more money, develop a large control experiment because we just don't have that much money.
We just kind of finishing up a chemical test with Dr. Dong Shah. And so he had some experience spraying ginger and heliconium.
Okay, yeah.
Got pretty good results.
Yeah, I like that. Yeah, I appreciated your talk.
So, I mean, if you need help, you have some resource, yeah,
Okay.
on the current work that Dr. Shah did.
Any reason why they thought this was a new disease? Apparently, this has been around for a long time. Is it weather? Is it culture? Is it?
So I think it's just that it got so severe in these areas because you see it all through Oahu. Once I started noticing it and started sampling for it, I could find it in isolated areas. The problem is that they let it get to a point that it was completely destroying the stand. So the virus was spreading enough through their stands of ginger that it was causing a problem.
Okay.
And it's something that I'm not necessarily sure if it's environmental because year after year they're seeing these symptoms. So it wasn't just one rainy season and their gingers suffered that year. It's these growers have had.
Well, initially we were told it's a new disease. Yeah, no, that's why I'm asking.
Well, there are some where they I mean, for example, the bean common mosaic virus, it had never been seen on ginger before.
Okay.
But we don't necessarily know if that's the main virus that is causing the decline symptoms because we have seen that the co-infection between BVRMV and CAYMV are causing symptoms. Yes, sir.
Did you guys notice if there was like a higher density of ladybugs when it passed that top?
So we haven't done any surveying in terms of that. I have noticed on plants that are infected have high populations of mealybugs, but I see clean plants with high populations of mealybugs too. On Oahu, for most of these, there's mealybugs all over.
But like for the plants that do have a lot of ladybugs that you see, do they have the virus in
They do, yeah. So there's plants that have mealybug, that have the virus, that have the mealybug, that don't have the virus, that have the virus, that don't have the mealybug. And there's no, yeah. But they could have had it at one point, yeah.
Have you guys tried like sequencing to see if like the virus DNA is present within the ladybugs?
So that's where we're at that right now. I want to work with John on collecting mealybugs and getting them tested, but just because it's in the mealybugs doesn't necessarily mean that they're vectoring it. It might be that they're sucking up sap and it's in their bodies, but they might not be going and vectoring it somewhere else. So we have to use them to vector it before we know that they are the vector.
Hey, Russell.
Yes, sir.
So do you know the vector of the bee common mosaic or the banana tree?
No, I don't know.
And how about nematodes? Do you know about the nematodes?
No work on nematodes either. So those are a pathway. Thank you, Brian.
Okay, thanks, Russell.
So that,
Very nice. Oh, do you have another question? Sorry. Another question.
if you have, as soon as you have something that we can actually send to the membership, if you could get it to Judy, and she'll get it to the board. If you wanna any kind, no, even prior to
Just a status
publication,
report.
Yeah. I'm gonna send
PowerPoint.
Judy. I'm gonna send Judy the PowerPoint.
something short and sweet where we
And
can
also,
get a link up.
I'm publishing into the Hanai, which is the sustainable farming newsletter of Oahu for soap, and I'll forward that to you guys. And that's on there is exactly what I've said here.
Thank you.